Thank you very much. If you talked a little bit faster, you could have put in a few more awards, but I'll take what I can get. I'm so happy to be here. I gave a lecture yesterday on the main campus. It was more for the general audience. And this one, although it's general in some ways, I'm trying to include much more data so you can see the actual technical proof of what I've been talking about over the years. And that's the formation of what's often called a dead zone. There are living animals in the surface. There are living bacteria in the sediments. But the name dead zone has been picked up by the press, shrimpers, and it's just hard to get rid of because that's how the public knows it. And I take advantage of it when I'm on radio or getting interviewed. But I'll go to oxygen depletion, low oxygen, hypoxia, which means hypo, low, oxia, oxygen, low oxygen. And I'm going to share in a somewhat whirlwind manner the research that we've been conducting over the years. I haven't done this by myself. I've had many collaborators. I started my first cruise 38 years ago. As I told the audience last night, I was 12 years old, so you can kind of do the math. And the watershed is huge. It's one of the largest rivers in the globe. It's with sediment, freshwater discharge, size, and area. It's a global river. It drains most of the continental U.S., about 40%, at least from the Rocky Mountains to the Appalachians into New York, Montana, Canada. And it delivers a lot of materials that reach the Gulf of Mexico. I consider it one ecosystem, the watershed and the coastal ocean of the northern Gulf of Mexico, which is linked to it. They're inseparable. What happens in the watershed happens in the Gulf. And the water from the Gulf ends up in the watershed. So we've got our own water cycle going on, plus nutrients and everything else. The definition is less than 2 milligrams per liter. It's a behavioral definition. Not everybody uses this one. They use 5, or they use whatever happens to be what the organism responds to, but we use 2 because at that level, if there are any swimmable fish or shrimp on the bottom, they leave the area. And a trawler will not catch anything when she puts her net over the side, and that's why it's called the dead zone. It's sometimes called dead water, red water, because associated with harmful algal blooms as well. The rays, like a stingray, I've seen flocks of stingrays up off the bottom swimming towards the shore to get out of the low oxygen at 3 milligrams per liter. And reproductive effects can happen at 5 milligrams per liter. So it just depends, but we stick with 2, and I've been pretty successful in making that stick a lot around the globe. Let's see. And it's worsening, of course, in global waters, and it's primarily from anthropogenic activities, which is excess nitrogen and phosphorus from a watershed that enters into the ambient water body. Hypoxia 101, this is a different figure from last night, so I decided I'd mix it up a little bit, but the fresh water comes in from the river. Here, and it forms a lens of fresh water, not fresh, it starts to mix over saltier water, and that generates a 2-layer system with a density barrier in between those two. So that's the physics. You have to have that density barrier because it keeps the oxygen that's being produced in the surface by phytoplankton from reaching the bottom. And the carbon that comes from the phytoplankton and the zooplankton fecal pellets all sink. Most of it sinks to the bottom. We've had particle traps out there before. and it contributes to the decomposition by the bacteria that uses up the oxygen because they're aerobic bacteria. There are also a lot of anaerobic bacteria out there when the system shifts from being oxic to anoxic. And if it goes below 2, it usually goes below 1. So it's not just a 2 milligram issue, it's a 1 milligram issue, or even anoxia, which is no oxygen. And you get hydrogen sulfide generated from the sediments and smells like a salt marsh, but you're in the Gulf of Mexico. This is the plume of the Mississippi River from Southwest Pass. What you see are the sediments. What you don't see is the fresh water, and you don't see the dissolved nutrients, the organic matter, nitrogen, nitrate, phosphate, silicate, and all kinds of trace metals and pollutants that come into the Gulf. But the diagram on the right has the main part of the watershed being above the confluence of the Ohio and the Mississippi, and that's where most of the water comes from. But the Missouri also is a source of not only water, but it's the main source of sediment over the years. So there are different contributions from the two, and most of the agricultural land is in the upper Mississippi and the Ohio River Valley, and rangeland, which generates phosphorus, is in Missouri watershed. It all ends up in Louisiana. Some of it goes through the main river, passes Baton Rouge, where I live. Some of it is often diverted through the Bonnet Carré Spillway. As this area knows, when floods, they let the water through, and you have water quality problems on this side of the river. And through the Southwest Pass of the Mississippi River. A third of the water is diverted through a river control structure and forms with the Red River, the Atchafalaya, and carries about a third of the total flow over the Mississippi. The simple story: more nutrients, more carbon flux, decreased water clarity, not from sediments, but from phytoplankton, and then decreased oxygen concentration. These are some of the first data from the area that were done systematically. This is the offshore, the Louisiana offshore and oil port facility, a deep water port that had a lot of really good baseline data. And you can see some of the earlier data showed that the oxygen was going down in the surface and in the bottom waters, and it eventually reached well below 2 before, a lot of low oxygen water, and that was in the Mississippi, that was in the Louisiana Bight off of Grand Isle, and it's now a major oil port with a pipeline into the rest of the US. At the same time, the sediments have been decreasing, but the turbidity is worse because of phytoplankton. So a lot of times the suspended sediments are not grains of sand or silt, they're phytoplankton. And the second disk depth has been getting less, which means the water's less clear because of phytoplankton, not because of the plume of the river. And we've documented that over a long time period as well. We do have some beautiful clear days, but it's called Old Muddy for a reason, and it's the muddy Mississippi, and that's what we love. So the Mississippi, as I mentioned, enters into the Gulf through the main delta and the Atchafalaya River Delta, which carries about a third. The dominant wind direction through most of the, winter, there's cold fronts, optimistic north winds, and then mostly southeastern winds from the southeast that push the water from the east to the west across the continental shelf of the Mississippi, of the Louisiana shelf. You can see sediment plumes here. And here, this is a MODIS image, but what you don't see are the dissolved nutrients. Phosphorus settles out pretty fast because it's usually adhered to sediment particles, but nitrogen is dissolved, and dissolved organic nitrogen gets into the microbial food web and it gets regenerated and reused, but the nitrate can also be taken up many times and regenerated and turned to ammonium and then turned back into nitrate and stimulate the growth of more phytoplankton. So the nitrate is everywhere on the shelf, primarily in the shadow of the two deltas, but it's everywhere. See what in the summer, the wind directions start to shift from the southwest or sometimes the west, and that pushes this water mass more to the east and piles it up against the Mississippi River Delta, and often pushes it around to the east side of the river into Mississippi and Alabama and as far as Florida at times. You have to have a really high flow for that to happen, but there is a movement of the water to the east. And what, keep looking at this, look at this. Mississippi, Mississippi. Okay. The most flow is in the spring. The lowest flow is in the summer. Most of the nutrients get to the shelf in the spring because of rainfall and precipitation in the watershed, but water still does continue to flow all the time because it's a massive river. It's a combination of physics and biology. You have to have a stratified water column that generates that two-layer system, sometimes 3 layers, from both temperature and salinity. And you can see from these figures over time that in May, where is that thing? The salinity, wait a minute, let me get this right. The salinity is lower on the surface and increases to the bottom. The temperature is lower on the surface and gets colder on the bottom. This pycnocline does most of the capping off of the low oxygen, but salinity is also important. And salinity is also, or whatever call it, salinity is also important, but the dissolved oxygen follows these density differences so that in a 20-meter water column, the lower 5, 10, sometimes 10 meters of water can be less than 2 milligrams per liter. It's not just at the bottom. As you move further away from the delta, it's more of a hugger of the bottom, but it does take up a huge amount of volume in the areas closer to the river. And it gets stronger in the summer. You can see here, the oxygen from 15 meters to the bottom 20 is close to 0. That's a lot of water that doesn't have any oxygen. And again, the temperature and the salinity form the density barrier. And then after a cold front or a thermal cooling, the whole thing turns over and you don't have stratified water anymore, except that you do because the salinity continues to be a major influence at the surface, not necessarily at the bottom. Same sort of a diagram, but this is if you took a slice of the continental shelf from Cocodrie to deep water offshore. We're looking through the water column here, and we're looking at density differences that are created by, get this straight, lower salinity, higher salinity, higher temperatures, lower temperatures in the summer, and then a decrease in the oxygen, in this particular case, from about 10 meters to 20 meters. And the density differences are shown here, and you can see a definite change in the density and the structure of the water column with one layer overlying the other. And then you can see that that density dictates where the low oxygen conditions are on the bottom, which in this case is this line right here. And if it's usually less than 2, it's less than 1, which means a pretty depleted oxygen scenario for the organisms that try to live there. Over the years, there have been changes in the nutrient loads to the Mississippi River. These are data from 1970. I'll show you some in a minute that go further back, but you can see that nitrogen in the form of nitrate has increased over time, and so has total nitrogen. The nitrate makes up about 30%, 30% of the total nitrogen. So when the nitrate goes up, the total nitrogen goes up. The phosphorus is not a very clear signal. Some people say it's doubled over time, but as far as I can see, there's not a pattern here. It's pretty flat. The discharge over time has also does not change much from the mean over time. So it's not more water causing stronger stratification. It does contribute to the stratification, but it's not getting worse because of more water. And then silica, which is important to the growth of diatoms, which are the base of the food web, have maintained a good level over the years. And the big change is, of course, in the nitrate. But the important thing is also the changes in the ratios of nitrogen to phosphorus and silicate to nitrogen, because the ratios of those chemicals, those nutrients, can dictate what kind of phytoplankton can grow. And if it's diatoms, it's good for the food web. If the diatoms aren't supported by the silica, then the other things that are non-silicious, such as dinoflagellates, which cause harmful algal blooms, can become part of the phytoplankton community. And we see that on the shelf. Longer term data, these go back to 1900. These go back to 1900 from a few stations. But you can see that the silica was pretty stable, and then it decreased from about 150 to it's now at about 25 micromolar. So it's dropped by a quarter. It's 4 times less now than it was historically. The nitrate is in the middle, and it has increased from historical levels, and it reached a peak, and now it's sort of leveled off and become more stable, but it hasn't changed. It's not going down with any management scenarios that are being applied. And the phosphate, again, pretty messy, but that's the way it is because it's adhered to sediments. But you can see a lot of variability in the phosphorus, and the record is not as long either. The changes in the nutrients are shown here over time since 1950. These are USGS data from Don Goolsby and Bob Mead, Old Man River, who's done most of the water quality work on the Mississippi over the years. You can see that the inputs from municipal and industrial are pretty level. Atmospheric ammonia is low, but also level. Atmospheric nitrate is also low and level over time. The manure has actually increased, but it's not evident because it's gone from free range to confined animal feed operations, which can cause a lot of problems. And there are more legumes being planted now, particularly soybeans, as a fertilizer alternative, cropping between corn and soybeans to fix nitrogen in the roots. And then the biggest change over time has been the use of fertilizer. And you can see it from the 60s, peaking in the late 70s, and now leveling off. And all of the paleo data that I'm going to show you follows the very same pattern, which indicates oxygen has gotten worse since the increase in the nutrients has occurred in the Mississippi River. see. We've also learned that the best relationship between what comes through the river and the size of the low oxygen area offshore is the May nitrate load. It can explain greater than 80% of the variability. Just a single variable. You can add phosphorus and get better. You can add discharge and get better, but that's in the load anyway. So there you can tick up the percentage explanation, but 80% just on May nitrate alone. And why May? For summer size, because it takes a while for the water to get to the Gulf of Mexico, for the phytoplankton to grow, for the fecal pellets and the senescent cells to sink to the bottom, and for the oxygen to be consumed, assuming it's stratified and there's no return of oxygen from the surface to the bottom. And you can see how well we have done in predicting the size based on the May nitrate load. It's a pretty close relationship. We're toying with increased water temperature now, which is also has a good relationship, and warmer water holds less oxygen. So that's one of the reasons we're looking at long-term. We published on in geologic record, a short-term record, but since the 60s, an increase in bottom water temperature. I think I may have a figure on that. We have two deltas. We have two regions that respond differently from the discharge. Most of the water is discharged, through the Mississippi River Delta. Is that button? And it can affect the whole area, but the influence is usually up to about where the Atchafalaya is. And these are data from nearshore to offshore. Our stations C1 to C9 in this area right here. And from January through December, I think this is a 4 or 5 year period average. And you can see that the low oxygen, it's not there in the winter because the water column is stirred up. It starts to develop in March, April, May. It becomes present in June, and you can see a large area, both in distance from shore and across the shelf, where the oxygen is less than 2 for extended periods of time. And then fall mixing and winter storms will break down the stratification. The other thing is that you can see the salinity signal as well, coming in with fresh water in the spring and having less fresh water in the late summer, in the fall and winter. And you can see the difference in the salinity from surface to bottom in this very same area. Same thing with nitrate. You can see a lot of nitrate coming in the spring, not as much in the summer, but there's sufficient carbon remaining in the sediments to continue the respiration that draws down the oxygen. And the chlorophyll peaks when the nitrate peaks, and you have massive phytoplankton blooms that support the fishery, support the zooplankton, but also contribute to the low oxygen on the bottom. If we shift to the other delta, this is the Atchafalaya in the center of the coast, carrying a third of the river. The dissolved oxygen there is shown here from near shore to offshore, from shallow water to deeper water. And you can see that the low oxygen is much restricted in time in an area compared to the figure that I just showed you. Still with mixing in the winter and the fall, but not as much at least in this area because of less water, less nutrients, less suspended sediments, lots of reasons why it would be different. The other aspect of this figure is the salinity. You can see peaks in decreases in salinity with spring flood, but also you can see a lot more in the summer as well. But you can see the definite lower salinity on the surface and higher salinity on the bottom. The nitrate, remember, it's a third of the river. There's not as much nitrate coming in here as there was in the previous slide for this area over here. And the nitrate is high earlier in the year, not quite as high in the spring. And then it's depleted, and not depleted, it's just, that's not much of a source of nitrate. But there's also in this area, the water travels across the Atchafalaya Bay and it gets regenerated as ammonium. And you can see that this is what's fueling the phytoplankton on that delta, not the nitrate. So the systems are very different, but they make up a total system that can be 22,000 square kilometers large. This is for Zombie DZ, but I don't see him in the audience. There you are. Yay. We have oxygen meters and we have them deployed at an offshore oil platform. We started with a single mooring with an anchor and a chain and a buoy, but we've had them at the near surface, middle and bottom, attached to an oil platform here. This is the famous Nancy station, C6B. There's Nancy changing out an oxygen meter, but you can see, and this is probably our best annual data for oxygen, continuous data, but you can see a cold front moved through. It mixed up the water column. Respiration drew down the oxygen, and this is April. So we have a lot of low oxygen in April. This is probably an intrusion of low oxygen water because it didn't last as long, but then a cold front moved through, mixed up the water, brought the oxygen down through respiration, so that the end of the month of July, which is usually when we're out there, is when we measure the shelf wide area. And then, let's see, it's May, June, hardly any oxygen on the bottom until a tropical storm comes through. And then respiration will again draw it down close to zero. And this particular year, the oxygen was close to zero the whole month of August, at least at this station. Sometimes deeper water that has oxygen intrudes into the area and it looks like it's reoxygenated, but then the system, the physics relaxes and this moves back offshore. So it's not a reoxygenation, it's a advection of water across the sensor on the bottom. And then in our favorite hurricane season, it gets lots of mixing, respiration to draw it down, phytoplankton again take over, and phytoplankton are pretty high for most of the rest of the year, just because there's continued phytoplankton production. This is our friend, the alligator, at our mooring station offshore in August after Hurricane Katrina. We went down. Adam did not particularly want to go down, seeing this thing. And when he got to the bottom, he really didn't want to go down because it's pitch black and our whole mooring was in pieces on the bottom. We knew that because side scan sonar had been used by LSU and we knew we had a mess down there. But he clung to me while we went down and went across the bottom. We couldn't see and found these pieces. This is a half meter and this is a whole meter. And then one of them was just completely gone. We don't know where it is, along with a nitrate and a silicate meter as well. But you can see the wind speeds picked up in time for the hurricane. The wave heights picked up, and you can see what was then a low oxygen area became well oxygenated, but it did go back to low oxygen. It didn't take very long because Katrina, for being a huge storm, was fast and small in the center, and it went right over our mooring, destroyed it, and then came into you all and destroyed you all. So that's that particular hurricane. We have lots of other data from hurricanes. These low oxygen areas affect the organisms that live on the bottom. You can have a massive fish kill similar to a Jubilee on the eastern Mobile shore. This is a Jubilee gone bad because it not only brought them close to the shore, but it killed them. Other things are stressed, trying to reach oxygen. This is not a place for an anemone, a Sarian theory anemone to live. It needs to be in the sediments. Otherwise, something will eat it, but there are no fish in there because there's no oxygen. So these things don't get eaten. And then there are also things that are dead. This is almost dead. And this is decaying. We call this our smoked crab. And Senator John Breaux saw this, and that's what got him interested in helping to pass legislation that funded harmful algal bloom and hypoxia research many years ago. He wanted to go to the dead zone and see the smoked crab. And it was October, there was no low oxygen, but he wanted to go diving and see it, but didn't work out. Plus it was too rough. It does affect fisheries, but there's no smoking gun. We still have high fisheries production in the Gulf of Mexico. You go out and want to catch something, you can catch it. But the poundage per decade average from NIMS data shows that it has declined, and all kinds of things are affected. The shrimp have to move somewhere else, or they don't have suitable habitat. They're more susceptible to predation. We had a particle trap out there once at mid-depth, and there was a shrimp sitting on top of it, adding particles to our particle trap. But it was up out of the low oxygen. We see things swimming at the surface that live in the sediments during the cruises that we're on. So it definitely reduces the habitat, makes things more susceptible to predation, changes their resources in the benthos, which basically get wiped out, and loss of diversity, biomass, abundance, and something else, species. There is one study that shows that the economics, whoops, I don't have it in here. The economics is the reason why the shrimp industry has suffered from low oxygen, because the juvenile browns come out of the estuaries, hit this mass, and stay there. And they're small, and the shrimpers catch lots of them, but they don't bring a high price at the market. Those that can get offshore outside of the low oxygen can grow to a larger size, but the economics of fuel and everything else and imports has caused the natural shrimp fishery to be wiped out, basically. And they're hanging on, barely hanging on. I'm not sure if I had my nitrawler slides, but subsidies for through the Farm Bill for the watershed, but no subsidies for shrimp in Louisiana. We do have harmful algal blooms also associated with this area. This is the year after the oil spill, and people were saying there's oil in the Gulf of Mexico, but this is a dinoflagellate, Heterosigma akashiwo. They've changed the name several times since, but it's a flagellate. It's toxic, and what you're seeing here is the bloom, and on the water, it looks like an oil spill. This actually is oil, but this looks like oil, and this looks like oil, but it was just thick, thick, thick with this particular phytoplankton, which was toxic. And fish don't eat it, and the fishermen get unhappy about it. This one came and went. Everybody said it's oil, so don't. No, it's oil. It's oil. It wasn't oil. It was phytoplankton. I think this speaks for itself. This is the cartoonist for the Baton Rouge Advocate, because we're now going into the dead zone. This is the area. It can be large, up to 22,000 square kilometers. It's from very near shore and 4 to 5 meters of water depth to 35, 45 offshore. It's right next to the beach, as close as the Pelican will let me. Ask the captain to take the Pelican to the shore, because this particular area here is very often hypoxic, but it's less than 5 meters deep. So the Pelicans, much as I plead, they won't let me go in there. Let's see. And it's also can reach 100 kilometers from shore, and it's most widespread in June, July, August, and September. Quick tour of hypoxia here. Oh, not yet. The particular area that I showed you was in 2011 because it was a flood year. We did get to the other side of the Delta and measured this area off of Mississippi, Alabama before we ran out of time and had to get the ship back. And I'm sure there's more of it. We know Dr. DZ is here and he knows there's plenty of low oxygen to the east of the river. And we're going to get some days this summer and next summer to go east of the river on the Pelican and map the same area that we've mapped before. But it's related to nitrate and discharge. It changes from year to year. 2012, 2011, 2018, 2019, go back. 2019, large area. I mean, the fact that this is less than, it's not in the line, it's just the contouring program, so it was a large area. This one was less discharge, but there are also winds in 2018 that pushed the water from the west towards the Delta and piled it up so that the footprint was small, but the volume was quite large. Another one here, a so-called typical low oxygen map, 2011, even in the flood year, 2012, less water. And this gives us this diagram, which is the size of the low oxygen over time. And I put letters on top of all the years if there was a drought, if the winds were from the south or the southwest, or there was a hurricane or a tropical storm. And so you can see the difference through the years, but very often the ones that are lower than average are due to the physics, not the lack of carbon in phytoplankton. Should I say what I said during the public lecture? I'll give it a go. ND stands for no data, but no damn dollars. And ND here means no data, but no damn ship. So there were no data in those years. We did get out in 88 and do other work. In June, there was low oxygen, but not when we took the shelf wide. If anybody wants to go on a cruise and get some cruising experience, stay in touch with me. I'm not in charge anymore, but the PI who is takes lots of students with her every year. And we have a great time. This is why the water gets stacked up to the east, because the winds prior to the cruise were primarily from the south and the southwest. And it forced the water mass and piled it up against the Mississippi Delta. In a more typical year, we see winds, we see winds from the southeast for much of the year, especially while we're out there, even some strong ones. And this may have had something to do with a smaller size that year. We had a hurricane in the middle of the BP oil spill that we had to wait to go out. So we went west first and then worked our way back. That gave us a very different map that year. But if you look at our success in using the May nitrate load, it's pretty high. And also the low oxygen area has gotten larger per unit nitrate load coming into the Gulf than it did historically. So it's an intensification of the low oxygen area as the nutrients have increased as well. I mentioned nitrogen, phosphorus, and silica. These are bioassays, test tube bioassays with phytoplankton and addition of nitrogen, phosphorus, silica, or the deletion of one. We do them in pairs. And we have 20 years worth of these data. And you can see that, I think you can see that, let me see. If you don't count these, because near the deltas, the phytoplankton are light limited. During the spring, June and August, they're both nitrogen and phosphorus limited. If you have only a single nutrient limiting, it's going to be phosphorus is much less to occur than the nitrogen. And you see that pattern in all of the ways we can group these data. So we talk nitrogen, we know phosphorus is important, but the main changer over time has been the nitrogen. And that's the focus of most of the remediation in the watershed, or attempted remediation. We do a lot of paleo work. We take box cores, we slice and dice, and we date them with lead-210 and cesium, and we look at changes in various factors over time that changed in these cores. And you can just see a short list here of changes in total carbon, more diatoms over time, more diatoms that are not as silicified because there's a silica limitation because of the high nitrate and the lower silica, changes in delta C13 and percent nitrogen over time, loss of diversity in foraminiferans, say that 4 times real fast. And ostracods, there's an increase in hypoxia tolerant foraminiferans, which are single-celled calcium carbonate individuals, protozoans that you can identify like a seashell and see how they've changed over time. Charlotte Bruner has done some work like this in Mississippi before. More intolerant forams, less tolerant forams. And these are all consistent with that increase in the nutrients in the 70s over time. And to make a few points, this is quinquiloculina. It's a foram, and you have to think backwards here. Oops. This graph goes from 1907 to present, and it was part of the paleo record until the late 1940s, and then it's not in there anymore. It's just not there because of low oxygen. This is quinquiloculina at another station, and it was high, and now it's variable, but it's not, it was low and it's variable. In this particular station in 27 meters, it was present until the 40s, and then it disappeared. So those are some examples of what the data look like. And these are other examples of foraminiferans in the top frame, either by what's called the AE ratio, which is the ratio of those that can take low oxygen over those that cannot. And that ratio has increased over time. This particular foraminiferan has increased over time. And so this is the foram diversity over time. This is glauconite is a clay mineral that is formed in reduced environments, which means no oxygen. And the diversity of the ostracods has also gone down. So you put all these pieces together and you can see the system is very different now than it was historically. The other one is a toxic phytoplankton diatom, Pseudonychia pseudodelicatissima. Love that word. Or a cohort of them. And even though the silica to nitrogen ratio has gone down, they're less silicified. So they've been able to take advantage of the high nitrate and respond with higher growth rates than they would have otherwise. And the competitors are not there because they can't keep up with the diatoms, and they're toxic and they cause amnesic shellfish poisoning. So, or death. And you can see that as the silicate to nitrate ratio has gone down and the nitrate flux has gone up, that the number of these toxic phytoplankton have increased in the cores, and that increase is also in the 60s to the 70s. It all fits together. It's a perfect picture. So when people tell me it's always been there, it has not always been there. It's prone to be there, but the paleo records tell us it has not always been there. We know where the nutrients come from mostly non-point source pollution. Point sources are very limited in the watershed, and of those point sources, it's mostly wastewater treatment. But then you look at the non-point source, which is 90% of the total load. The nitrogen is well over 50% corn and soybean rotation and other crops. And the phosphorus, because of the manure, is about 37% of the load of phosphorus. So different mitigation activities can approach both of these, but they both need to be approached in a harmonized way without causing more problems. The watershed has also changed. I'm getting close to time here. And I wanted to show you this because it's not just the Gulf of Mexico. The watershed has changed tremendously over time. The number of virgin forests in the top panel has decreased to almost none nowadays. In the upper right panel, the wetlands lost in Illinois, Missouri, Indiana, Iowa, prairie potholes, they've been lost. And you can see in the bottom right that the amount of nitrate that is held within the marshes is higher when there's more marsh. We've cleared the land. We created a dust bowl. The tree that's sitting on that promontory is sitting on eroded land in the Tennessee River Valley. And the landscape is very different now than it was before. We'll never go back to the same landscape. All of these states have had major decreases in grasslands, prairie grasslands that have longer roots that can handle nitrogen overload better than a corn plant, which has very shallow roots. The Mississippi River has been leveed so that the overflow into the adjoining floodway is not happening anymore. And that's a natural way for denitrification to remove nitrogen. And of course, we have major shipping channels that need to be maintained. Let's see if I've got any more. Whoops. Wait a minute. The roots on the left are from wheat. A corn plant has even fewer roots. But a blue stem grass in a prairie has exceedingly long roots. We used to have them hanging from the roof in a hallway at LSU. But you can see that where there's bluegrass native cover, there's much less nitrogen that comes off of the land and other types of activities. And plowed croplands release a lot of nitrogen, and those that are plowed and drained with plastic pipes are also a high source of nitrogen. The rivers have been dammed. What used to be high flux of sediment to build deltas is no longer there. This is the work of Mississippi River Muddy, Bob Mead. And you can see these are guesses for some of them, but the the sediments that are coming into the Gulf are now 50% less than they were before. And it's not due to discharge, which is this panel, which is, it's variable, but it's even over time. The landscapes in the Mississippi River watershed are compromised. There are many places where nitrate levels are above drinking water standards, and that causes toxics and problems with what's called blue babies and cancer. And these harmful algal blooms that get generated just like they were here in Mississippi Sound when the Mississippi River was sent your way are cyanobacteria, and they are toxic. And if a pet falls in it, they're going to die. There have been people who have fallen in it and they've died. So it's not good. And this is in the watershed. So fix your watershed will also benefit. These are the factors that are important in hypoxia, but I've talked about those that are. But I just want to say that a deeper water oxygen minimum layer does not come up onto the shelf. The river carbon does not play a role in the respiration. It's all in situ produced carbon from the surface waters. Wetland erosion has very little of an effect. Estuarine nutrients, very little of effect. The levying of the river definitely has an effect, but Missouri is where the suspended sediments fell out. Upwell nutrients aren't an issue, and climate not as yet, but we have data that shows that the bottom water temperature has increased over time in the area below oxygen, and that means less oxygen dissolved in those warmer waters. So we're keeping our eye on temperature right now. If you look at, what time is it? I'm going too long. Yes, I am. This is my modules of climate change with increased precipitation leading to more water, and along with nutrients, productivity, vertical carbon flux, ending up in bottom water hypoxia or possibly harmful algal blooms, and warming waters, sea level rise, water temperature, winds can be affected. The metabolic processes are increased up to a point, but nutrients are still limiting, and the physical environment can be changed as well by more warmer waters or more rainfall or lots of other things. So we have an unknown climate in our future as well. There are many of these around the world. Pay attention to the red dots. This is a paper that Denise Breitberg did. She's the lead. I'm one of it. I'm one of them, and it was in Science, but there are natural areas of low oxygen that go through the same processes, but it's these coastal areas that have become hypoxic that they weren't historically. And I think one more. I gave a talk in on the Rio Paraná many years ago, and this was in November of 1999, and they told me that their rivers in South America were too large to have problems. And all those red dots now show where there's hypoxic areas along the South American coast, not the Amazon. The Amazon has huge currents that carry the water away from the coastal area. So there's no residence time for the phytoplankton to grow. And the future, we need to take care of things. I have snake oil salesmen come to me with solutions for hypoxia. And these are some of them. Just put a bunch of aerators out there like an aquarium, or put this mysterious chemical out there and it'll fix low oxygen. But there are problems with all of them. The volume is too large for any mechanical fix and probably any chemical fix. I think that's it. Yeah. So thank you for being here and letting me show all of my data. Nobody left. So thank you very much.